Welcome everyone to OsteoBites and happy Thursday. And we are very thrilled today to have Corrie and Taylor as our guests today on OsteoBites. I'm Christina Iptoma. I am mom to Osteo Angel Dillon and the Director of Scientific Programs for MIB Agents. And we have a great conversation today about the Osteosarcoma Project. So I wanted to introduce Corrie. She is a Deputy Director of Count Me In, and she's a research scientist at the Broad Institute of MIT and Harvard. She is a trained cancer researcher with a Ph.D. in biochemistry, and she completed her postdoctoral work in cancer immunology, focused on melanoma. In 2010, she was diagnosed with angiosarcoma, and she has combined her cancer advocacy and scientific program to engage with patients in order to build and carry out patient-partnered genomic studies. And she's also the co-founder of Angiosarcoma Awareness Inc. And Taylor Cusher is a clinical project manager with Count Me In at the Broad. In this role, she manages several patient-partnered research studies that generate and share clinical, genomic, and patient-reported data for rare cancers. Prior to joining the Broad Institute, Cusher led programs and patient access to health data and patient engagement in medical R&D at FasterCures, a center of the Milken Institute. And in these roles, her passion for enhancing connection and community at the intersection of patients and research is at the forefront of her work. So welcome both of you today. Before we get started, I just have a few announcements. The first is Factor registration is open, and I'm going to put the registration link in the chat. It's June 23rd to 25th in San Diego, and I think Taylor and Corrie are going to be there, right? Yeah. So there's incentive enough to come. And if osteosarcoma is your jam, this is the place to be. It is the only osteosarcoma conference held in the US, and we have something for everyone in our community of research scientists, clinicians, patients, caregivers, and bereaved families. We will have incredible speakers on 8 scientific panels, including Corrie. And we are going to have an Osteo Warrior HQ that we have every year, which is free to Osteo Warriors and siblings 18 and under. It's a great place for them to connect and hang out. We're going to have wellness programming this year, which is new, which we're really excited about. It's available to all registered attendees. If you guys are going to sneak away from the scientific panels, and we think will be of particular interest to our bereaved families. And while we're all drawn together there for a pretty sucky reason, it is an amazing community, and we all manage to have a pretty good time together. So we hope you guys can all come. And for any clinicians joining us today, MIB Agents hosts a bi-monthly virtual tumor board for osteosarcoma called Turbo. The next one is Wednesday. It's coming up. It's May 11th at 5 Eastern. And if you'd like to be added to the calendar invite or have a case to submit, please let us know. I'm going to put another link in the chat where you can just sign up to get more information or be added to the calendar invite. And lastly, I wanted to announce our new AYA podcast, Osteo. So last week we did not have our regular OsteoBites, and instead we dropped a new podcast by 4 members of our junior advisory board, Camille, Kerrigan, Vicky, and Mia, and they spill the tea on all things osteo and talk about their experiences living through osteosarcoma. So tell your kids about it, tell your patients about it. You can find it on our website and on your favorite podcast platform, and we'll put a link to the podcast in the chat as well. And I would like to thank the sponsor of this episode, BTG Specialty Pharmaceuticals. BTG provides rescue medicines typically used in emergency rooms and intensive care units to treat patients for whom there are limited treatment options. They're dedicated to delivering quality medicines that make a real difference to patients and their families through the development, manufacture, and commercialization of pharmaceutical products. Their current portfolio of antidotes, cataracts, certain snake venoms, and the toxicity associated with some heart and cancer medications. Their drug Roxase is for high-dose methotrexate toxicity. So thank you to our sponsor, BTG. And with that, I'm going to hand it over to Taylor and Corrie to go ahead and get started.
Wonderful. Thank you so much. Taylor's going to share slides for both of us, but I'll kick it off. So thanks so much for joining us today. We're going to talk about something we've talked about several times as guests on OsteoBites, which is our initiative out of the Broad Institute's Count Me In program called the Osteosarcoma Project. And there's some really interesting updates that we're bringing to you today. And we hope that you have lots of questions that we can talk about. So please, let us know, and know that we are always happy and willing to talk with any stakeholder in the osteosarcoma community. That is what we hope for more than anything else in the lead up to generating and sharing data, is that we can generate and share as a result of deep engagement with each and every one of you. So today we're going to talk, I'm going to give a little bit of background on just Count Me In, like how did this even come to be? What is, where does Osteosarcoma Project fit in? And then I'm going to hand it over to Taylor, who's going to talk about new features that are coming online in 2022. So a little word about Count Me In just in general. I came to the Broad back in 2015 to help the leadership of the Broad Institute. And actually, I'm going to start with what is the Broad Institute? The Broad Institute is this really incredible research facility in Cambridge, Massachusetts. Its full name is the Broad Institute of MIT and Harvard. And it is, it's where the human genome was sequenced the very first time, and where they do massive, huge scale genomics research. And it's world renowned in terms of the types of discoveries that are made and the types of collaborative research that they support and sponsor. And it's such a privilege to work there. And I came there after I did a postdoc in cancer immunology, because I was really interested in figuring out how we could change the system of cancer research so that those of us with super rare cancers like osteosarcoma and angiosarcoma, there's only about 300 people a year that get my kind of cancer, very similar. I came to just try to see, could we leverage this incredible infrastructure to come to bear in a meaningful way for those of us with super rare cancers. And I came really because the leadership of the Broad was, they were asking these questions. They were asking, what if we could generate a massive publicly available database of clinical, genomic, molecular, and patient-reported data in cancer to enable researchers to find patterns in the data and help accelerate discoveries in new development of the development of new treatment strategies. And I thought that's really incredible. The way that cancer research happens for the most part is that, at least for adults, that you will have some really well-funded major academic medical centers, and that's where almost all of the research gets done. So if you're treated at a smaller center or in a community setting, which is not as typical in pediatric cancers, but it is in adult cancers, the chance of your tumor or your experience going to inform the research world is slim to none. And I did not appreciate this until I was diagnosed myself. And when I was diagnosed and I went inside of one of these major academic medical centers, the information that they were collecting wasn't I couldn't believe what they weren't asking me. I couldn't believe it. They were what's your name? How old are you? What's your weight? And I said, well, don't you want to know my smoking history? Don't you know all this other stuff? And they looked at me and they said, well, we don't think that there's a connection between that and your cancer. And I said, well, has anybody studied that? And they're like, no, because we don't think there's a connection. And I was like, wait a minute, something's wrong here. How do you know if you haven't collected the data? And how, and how do you even know what data to collect if you haven't asked any questions? And that got me to thinking, maybe I need to peel this onion back a little bit. And when I did, I came to really understand just how little data is actually collected and studied by scientists everywhere. So when I saw this opportunity, I was like, oh, that's it right there. And I was like, I don't even care what cancer they're, not that I don't care. Everybody with cancer deserves better treatments no matter what. And so I was cancer agnostic, I guess is a better way to say, in terms of where we started, because I saw that if we could make this successful, it could absolutely be pointed to rare cancers. And if we could point this to rare cancers, we really had a shot at generating massive data sets where nobody else possibly could for all of the reasons that I'm sure all of us are familiar with. No money, patients are scattered. There's not enough of us to study. There's not enough money for researchers who are even interested to like make a bona fide career at scale. And so this was like a great way to like overcome all of those things. I'll go like this one. I want you to, there we go. And so we went ahead and built the Metastatic Breast Cancer Project. This is where we started. And we started in metastatic breast cancer for a number of reasons. The director of Count Me In is Nick Wagley. Dr. Nick Wagley is he's a clinician at the Dana-Farber Cancer Institute. He treats metastatic breast cancer patients, and he's a genomicist at the Broad Institute. And so he kind of like spans both. And he really felt strongly that there was tremendous advocacy and engagement of patients with metastatic breast cancer. And it was a fairly large patient population for us to start with. And so I started in this disease community, and I did not understand or appreciate until I started deeply working with the patients just how similar experiences are regardless of the cancer type. And it was such a privilege to work with the hundreds to thousands of men and women that I've met over the years to build, launch, and oversee the Metastatic Breast Cancer Project. It's just very near and dear to my heart. And so it's just like such a great honor to know that we have over 6,000 women and men who have joined, which is the largest cohort of metastatic breast cancer patients ever assembled. We have patients from all 50 states. And if you look at it, it looks like they're all kind of like on the East Coast. But if you overlay like the population graph of the United States, it's this exactly. So this is just where humans are. And so because this is where humans are, that's also where they're going, where they live when they have cancer. And so if you then look to the right at the Angiosarcoma Project, again, this is such a rare cancer, just like osteosarcoma. And there's like one Facebook group. That's where I know all of the angiosarcoma people. There's like not enough there's not enough of us to support like a bunch of like robust support groups. And so people will, and this may resonate, but people will start other groups or they'll like go other places. And there's enough of us kind of scattered about that when we see that happen, we're like, that's cool, come here too. And so everybody kind of funnels into the same place. And that's where we reach the vast majority of people in my disease community to tell them about this project, which we launched in 2017. We have over 640 women and men that have angiosarcoma that are part of this project, which is a huge number when you have something this rare. And we also have the capability of enabling loved ones to provide information about their loved one, like their children or their parents who passed from this disease. And we've learned quite a bit, and it's made me realize, and I know that there's a couple people in the osteosarcoma world that we've been working with closely to really dig in with what we can do to work with like bereaved parents and loved ones, because I don't know a group of people who's more familiar with the cancer experience than a bereaved parent, and that they can be transformative. And so many people want so much to help so that other people don't have to experience the tremendous pain that they have. And so this was all like part of that was launching that in angiosarcoma and then really building it out with the osteosarcoma community. So more on that, in another talk, but just wanted to highlight it.
And that can be a super empowering experience to be able to share that data to help others. And I'm curious, Corrie, when looking at these maps, you had mentioned trying to get more representation from some of the smaller local community centers. And so when you look at these maps, at least since some of these have been going on for several years, have you found that you've been getting good representation outside the major academic institutions?
Yeah, that's it. So rural representation is one of our sweet spots where we do very, very well. And so we do, although in angiosarcoma is so rare, we don't have people from like the top middle of the country, so I don't know if people are just not getting angiosarcoma there or if they haven't heard about the project, but it also took us, Montana was the last state for us to have somebody enrolled in the Metastatic Breast Cancer Project too. But I think we have looked, and we absolutely have over enrollment of patients that are in rural settings relative to like the big settings that we have where the like traditional research is.
That's great.
Yeah.
And I'll just I'll add that when patients sign up for the projects, they tell us all of the places that they have received treatment or care for their cancer, and we make requests from all of those institutions. So I know in the Metastatic Breast Cancer Project, or maybe Count Me In as a whole, we've made requests for over 1,200 different institutions across the country, the United States and Canada, in our time. So we're getting records from every corner of the US and Canada where patients have been seen and treated.
It's so cool too, when you look at that graph, like those 1,200 institutions, they only have one patient participating. So if you think about it like from like a study design, there's no way you can build a study and do a collaboration among 1,200 institutions, all the paperwork that you'd have to do. But patients, the power of the patient, patient will, and honoring the patient will to participate is what makes this totally possible and feasible. So it's a very cool kind of infrastructure that has been put in place. Mm-hmm. So what we do is we generate clinically annotated whole exome sequencing and RNA-seq, which I'll talk about in just a second. And when we take information out of the medical record without any of the identifiable information, we connect them, and then we put them out into the world for free wherever we can. So we have it in a place called CBio Portal, which is when I was a postdoc and I wanted to know anything about like whatever gene I was studying, this is where I would go. This is where all of the postdocs go. You know, it's just where people go when you're in science to like try to figure out what's going on with your gene of interest. And so it's just kind of like baked into the culture of science. And so that's one of the first places we went. And that's for the like processed data. So that is for non-computational biologists to consume, use, and make discoveries off of. We also house all of the raw data in a more kind of like secure database in the National Cancer Institute's Genomic Data Commons. And this is for like the genomics wizards that know how you can actually do the bioinformatics and really dig into the raw data and process and learn about it from scratch. I have a tremendous amount of respect for those humans. I don't have that capability, and I'm always impressed when they do. And then we also have, homegrown databases like Terra and patient researcher specific portals that we house the data. The whole, back to like the whole exome sequencing and RNA-seq, for anybody listening that doesn't have kind of a background in genomics or science, you can think of the exome as all of the parts of your DNA that can be turned into protein. And when you have cancer, it's the proteins kind of going crazy that really lead to cancer being this like awful disease. And so when we look at the parts of the DNA, which are called the exome portions, we can literally read the genetic code and what kind of mutations are happening in somebody's tumor versus like their normal DNA. And that will often lead us to understand how that code changes the structure of the proteins and makes them act differently. Sometimes those changes make a protein that's supposed, like proteins all move. That's the thing, like they're three-dimensional structures that move all the time. And so maybe one of the proteins needs to go like this in order to function. And it only does that when it has like calcium near it. But if it has a mutation, it does it all the time. And that action might keep the cell like going and splitting and dividing without the ability to stop. And things like that actually cause cancer and drive cancer. And that's what we're looking for when we actually do that sequencing, are those changes and what the result happens. RNA is going from that genetic code in the DNA. It's like literally making a copy of it to go turn into the protein. And so we read both of those types of sequencing in order to better understand. This is a current snapshot of where we are with these two most mature projects. In the Metastatic Breast Cancer Project, we have released data on 400 samples from 300 patients. Again, the samples are whole exome sequencing and RNA-seq. There are like over 30 papers that other scientists have written and gotten through peer review, making discoveries on that data. We have a manuscript in preparation, and we've been able to translate the whole project into Spanish. In angiosarcoma, we've published our own discoveries in a prestigious journal called Nature Medicine. There are like a bunch of clinical trials that are validating all of these discoveries. I got a text from a patient on one of these clinical trials about 2 weeks ago that just brought me to tears. And she was saying that she was metastatic, and she went on this clinical trial that the doctors designed based off of our data. And she said, I am doing extraordinarily well. Every single time I go in and get scanned, the doctors are just like totally in disbelief because it's just, gone away. And she said that she thinks that, or the doctor said that if the trial continues this way, it will change standard of care for angiosarcoma patients. And so it's like true impact. And that's kind of in the rare cancers where there's just not a whole lot done to like move the needle. If we come together like this and we can get all of this data in the right hands, it could be transformative like that and lead to new treatments. I'm like seeing it happen in real time, and it's really inspiring. And I'm really hoping for the same for osteosarcoma. So we have, there are also multiple papers by independent researchers, and we have data in the public domain, 83 samples from 62 patients. And we also have an analysis underway with like 200 healthy normal, or not healthy normal, but like normal DNA samples from angiosarcoma patients, so that we can look at the inherited or germline DNA to see, are people born with like a predisposition for angiosarcoma? If so, then what, right? Like, do we get screened more? Like, what do we do for our children? Things like that. And so that's also on its way. So we launched several independent projects, like MBC Project, Angiosarcoma Project, Metastatic Prostate Cancer Project, Brain Tumor Project, Gastroesophageal Cancer Project, and of course, the Osteosarcoma Project. And then we were able to open the platform to everybody with cancer in the United States or Canada, regardless of age. And so this is where everybody lives, and all the different colors are people that are joining from these different individual projects. But now I don't know that it's in this presentation, but it's super cool. We have participants in Count Me In that have over 100 different types of cancer, which is just remarkable. And it just provides such an opportunity for us to learn. But back to osteosarcoma, I want to show this beautiful picture of one of our friends here.
I recognize his cheeks.
Yes, near and dear to all of our hearts, of course. It was such an honor to work with him when he came to visit us. This is a copy, or this is a picture of our website, osproject.org. And you can, anybody who wants to join the project can just go to this website and click on Count Me In and join. We launched this. I actually launched it at Factor in 2020. And since then, we've had 258 patients that are registered, which is awesome. We have 82 saliva samples that have been received and actually are undergoing sequencing right now. So that same kind of like look at the saliva to try to understand what might be, are there predispositions that we can learn about? That's underway right now. We have 230 medical records that are requested, and we're working on tissue and blood samples as we speak. And there's actually, we would be further along with this project, but we went ahead and got a really massive grant to expand it. And that has caused us to do a couple of things, like stall a little bit so that we can kind of rebuild everything from the inside, so that instead of just research grade data, we can generate data that can actually be shared with participants in the project. And Taylor's going to go into all of those details, but that process is, I don't want it lost on anybody how much detail went in and is still going into looking at every single thing we do to make sure that it is regulatory, has the right regulatory components that will enable us to share. And there's like a whole lot of stuff that goes into that, but it has been such a tremendous turning point for all of us that work at Count Me In. This is something we've all wanted for a very long time. And it's such a sweet thing for it to be coming to life in the Osteosarcoma Project. So we're all very proud of it. We're excited to launch that part of it.
So much great stuff going on. So, your last slide kind of got me thinking that, first of all, you have a huge database of the metastatic breast cancer, but with all these different types of cancers. And, this has come up because MYC is being studied a lot for osteo, right? And, but MYC is common in lots of other cancers.
Yeah.
And so, as we kind of move from this, versus it being kind of like a site-specific type of tumor to kind of more of a molecular target type of tumor, it seems like all that information you're collecting on the other cancers, you'll have this kind of unique opportunity to then kind of filter it, not so much by like the type of cancer, but just what some of the molecular targets are. And so
oh yeah, I could not agree more. I have there's so much there in what you just said that's so important that I think is going to lead to so much understanding about cancer that we haven't had at the ready, because you're right, everything's like, it's a breast cancer, so only the breast cancer people study it over here, and it's a sarcoma, so only, but there's so much to learn along those lines. Just take MYC, for example. MYC is also way amplified in a subset of angiosarcoma patients. And there's two groups of angiosarcoma patients, even you could further refine. One is like people that have sun-induced radiation caused endothelial cell angiosarcoma, like in their skin exposed areas. And then the other is radiation associated angiosarcoma. After women get breast cancer conservation therapy, they almost exclusively have MYC amplification. And so it's really interesting. And then there's of course, all the other cancers, but in sarcoma, when you take a sarcoma, because sarcomas can arise kind of anywhere and travel anywhere. They're not necessarily the same as a carcinoma that is like specific to one tissue or one environment when they start. Like and I'll say, like in angiosarcoma, I had angiosarcoma in my breast, but not breast cancer. It was like an endothelial cell cancer that happened to be in that microenvironment. And so you can, and then when we did the study, it turns out that breast angiosarcomas, almost like unlike any of the angiosarcomas that go anywhere, like pop up anywhere else in the body, about 30% of them had mutations in a gene called PIK3CA, but none of the other angiosarcomas had them. PIK3CA is one of the most commonly mutated genes in breast cancer. And so that's like, it blows my mind. Like there may be niche specific selection of cells that have mutations for PIK3CA because of the microenvironment. Nobody would know to even like put those things together if you didn't have a totally independent cancer that just happened to be in the breast at that time. But it's, so anyway, just to your point, we will be able to study all of the different cancers based on the molecular underpinnings, and then trace that back and maybe learn about like cancers just writ large as a result of the data that we're generating. So yeah, agree.
Yeah.
So our key areas of impact, and this was drafted from Katie Janeway and Brian Crompton. And so they're really leading the charge on the science here, and they're really incredible to work with, as I'm sure everybody here knows, and everybody in the osteosarcoma community probably is well aware. They're just stellar humans and so smart and so dedicated. And it's been such a joy to like work with them behind the scenes and just see that dedication and that mission and that passion at every single turn. And so they've really identified three kind of buckets that are key areas for impact. One is to further define the molecular characterization of the subtypes. So osteosarcoma, like many cancers, is not just one thing. And when you really dig into the DNA and really start to understand, they can be broken into about 6 different genetic subtypes, but we don't know a whole lot about each of them. And so this project, if we can enroll enough people, we can really study that at scale. We can also learn about tumor evolution. So what happens, from an early stage tumor as it progresses, what happens over time throughout treatment? How does the tumor change over time as it becomes metastatic? And if we can learn all of those different points of change, maybe we can target them and find different, better therapies at each of those different transition points. And that comes down to the final point, which is the identification of new targetable therapies. If we can combine all of this deep knowledge from these first 2 buckets, there could be opportunity to either like find drugs that already exist that we think, ooh, now that we know all of this, we should be pointing these drugs over this way at this time in the progression of the disease, or maybe it will give us insight into what drugs we could just make from scratch.
And just curious, Corrie, based on the experience also from other, I know it's kind of still early for the Osteosarcoma Project, but based on the other, like NBC, have you seen success in getting kind of more of that longitudinal data. So once someone gives you access to their medical records, or, once at least you connect with that patient, that then you're able to kind of get that information over time, which it does give you a lot kind of richer data to work with.
Yeah, totally. So we do, and one of the biggest ways I think that we build longitudinally is through follow-up surveys. Mm-hmm. And so in the Metastatic Breast Cancer Project, we built a survey to get updates from patients and deployed it like 3 years after the launch of the project, and over 1,000 people sent them back. It was incredible. And so we have the capability of collecting additional blood biopsies over time. We can do updates to the medical records. We don't have it like a systematic way to continuously update, but I think what we were more likely to do is when we'll pick like cohorts of people that we want to continue to get updates and follow them and request more records over time. And we have done that, like in angiosarcoma, when we were getting ready to publish this paper, we wanted updated records for the people who had these extraordinary responses to a certain therapy to say, are they still doing well? And we were able to kind of confirm over time that they indeed were. And so for sure, we have that capability, and we do leverage it from time to time, just not necessarily at scale across the whole cohort. So our goal for the Osteosarcoma Project is to generate clinically annotated genomic data sets. So that whole exome sequencing, whole genome sequencing. So real quick, the exome, the part that turns into the protein, right? But then there's like so much other DNA, and like nobody knows what it does. There's like so much DNA. There's DNA that like regulates the whole process. There's DNA that, like, if it's open or if it's shut or if it's wrapped around a nucleosome, will act differently. We don't know what any of it does. And so like scientists are really just starting to figure out what roles do the rest of the DNA, what do they come to bear on cancer. And so we are able to do the whole genome sequencing to learn about that, but also to learn at like big structural events that happen within the genome. Like in osteosarcoma, there's the actual architecture of the chromosomes can take on a different look, and that in and of itself can be informative. And so doing the whole genome gives us like all kinds of different complementary information in the whole genome sequencing. And of course, that RNA-seq, which is like the copies of the whole exomes, that gives us a really good indication of like how often is that gene either totally normal or mutated, like how often is it copied? If it's copied one time, you get one protein, right? If it's copied 10 times, you get 10 times the amount of protein. And so sometimes you just get extra copies of a protein that can cause that cell to go crazy. And that's also contributing to cancer. And so all of these different sequencing metrics are measuring different but complementary things that can all inform on our understanding of on osteosarcoma. And then the big one that Taylor's going to take over here in a second and go through is sharing these learning. Like, what are we learning? If we can learn it, you should learn it too. We're not like a clinical genomic sequencing vendor like you might hear about, where your doctor orders a test and you get information back 3 weeks later. We're a research project, but we feel like the philosophy is if we can see what's going on and learn, you should be able to see and understand the same stuff that we can see about your tumors. And so that's kind of like the big thing that we're working on now. And she'll go over our shared learnings from tumor sequencing and germline. And also we are studying how we engage patients so that we can do better at it. And also collaborate, and we want to collaborate and share with other patient engagement projects. And with that, I will pass it over to Taylor. Thanks, everybody.
Great, thank you, Corrie. So before we get into what's new and different on coming to the Osteo Project, I wanted to spend a little bit of time talking about what happens with samples and information provided to the project, because that, what is it that we're doing when we ask ask participants, where have you been treated and what do we do with that information? So just talk through that really quick first. So there's a variety of different sample types and information types that we receive from osteosarcoma participants, and thanks to participation in the Osteosarcoma Project. So saliva samples, which we send a saliva collection kit to everyone who enrolls in the project. These samples can tell us about inherited or germline DNA, and these are also a necessary component for us to be able to sequence tumor samples as well, because when the tumor samples go to the sequencers. They have to be paired with this germline sample so that we can then, during the analysis phase, understand what components of the exome are coming from that inherited DNA and what components of the exome are just in the tumor, so that we can zoom in and see what are those changes that are specific to the tumors themselves. We also have participant surveys for folks in the project, and this helps us understand who's participating, collect some demographic information, where people live, when were they diagnosed, what's their cancer experience been like, and much more. And this is also something that we will, as Corrie mentioned before, will continue to use surveys as a way of engaging folks and learning from them more throughout their time in the project. We also request medical records from institutions where folks have been treated. And in getting back these medical records, it helps to provide our team with a detailed history of all of the procedures, all of the treatments, and a lot of other valuable clinical data points that we use both internally for helping to identify cohorts and figuring out which tissue samples we might be able to get for participants, but then also this clinical information becomes a component of that data that we share and make publicly available. We also have the option for collecting blood samples from participants in the project. And so this blood samples can also be used as a source of inherited or germline DNA. But we also take a look at blood samples to see if there are clues in understanding tumor DNA that actually makes its way out of the tumor cells and circulates into the bloodstream. And so for blood samples we receive, we'll take a look at those and do something called ultra low pass whole genome sequencing to get a sense of how much tumor DNA is in the blood and is there enough for us to sequence it and see, learn a little bit more about those, the DNA of the tumors, maybe in the absence of a tissue sample or alongside a tissue sample. And last but not least, tissue samples, of course, provide us with samples of the DNA from the tumors themselves. And we pair those, like I said before, with a source of a germline or a normal DNA when we're sending things off to the sequencers. And this is really where we'll be able to dig in a little bit more, understand those subtypes, tumor evolution, potential targets, and those lofty goals that we have for the project that Corrie outlined earlier. So all of these are really essential and important elements of information and samples that we get from participants. And so we are so grateful when folks, give us any one of these, but especially the depth of information that we can get on a single participant by having a collection of different samples, different surveys, different medical records, really will truly make an impact to see, longitudinally, how has cancer progressed, and have a bunch of data points for folks to be able to query and hopefully answer some questions about osteosarcoma with. So finally, turning to the piece that we have been previewing throughout. So additional and new and exciting opportunities are coming to the OS project in the next couple of months. And so this comes with sort of in three main categories: learning more about your tumor DNA, learning more about your normal or that germline and inherited DNA, and also responding to more surveys and having more opportunities to engage with the team. So I'm going to talk through each of these and what they look like. Also know that when we do release these and make these part of the Osteosarcoma Project, there will be a lot of new information on the website. It goes into detail about all of these things, FAQs, but our team is always here to answer questions that anyone has about participation. So this will not be your only opportunity to hear this information for sure. So the first is learning about information from tumor samples. So as Corrie mentioned, we believe and have always wanted to be able to share back with participants things that we are learning from the tumor samples, so that, just to be transparent and say, if we know it, then you should know it too. It's about, your body and the sample that you have graciously provided to us. So if participants choose to share tumor samples with the project and we're able to successfully sequence those, we'll be able to generate a letter about what we're learning from that tumor sample. So it might highlight some of the genomics and say, we did analysis, and we were able to find, variants in these 4 genes in your tumor. And this is what we know about these genes, and this is what we don't know about these genes. And given that osteosarcoma is rare and there's much research that needs to happen, most of these, it may be very unclear what these variations in the genes mean. So it's really intended to highlight how samples from participants are contributing to research and helping us learn and kind of contribute to what are these gaps in research right now. And most likely will not, this information that we're returning will likely not have implications for treatment or prognosis at this time, simply because those research gaps still exist. And I will also mention, because of regulatory restrictions, which we can talk about in more detail if folks have questions, this will be unavailable to participants in New York State and Canada because of regulatory guidelines and challenges. A lot of bureaucratic red tape, as you can probably imagine. So the other thing that folks can learn more about, thanks to contribution of their samples, is this normal or inherited or germline DNA. So after submitting an initial germline sample to the Broad, participants will be provided with an option to undergo germline testing with a group that we've partnered with called Invitae. And participants will be able to provide an additional saliva sample directly to Invitae and complete a couple of telehealth appointments with Invitae to understand what it is that this germline panel test will go through, and talk through the testing, what it means, what it may mean for the patient or their family members, and then have this panel test run, which will look at 84 specific genes associated with different types of cancer. Those results of that panel will be delivered directly back to the participant or the parent, depending on the age of the participant, through a post-test telehealth appointment with the genetic counselor, who will also be able to explain, answer questions, and talk about the implications of any of the results of that panel test.
That's really cool. And there's no cost to any of this, is that right, for participating patients?
That is correct. Absolutely no cost to participants. It is all entirely on the project.
And what is the turnaround time for getting results on something like this?
Yeah, so the turnaround time for this, because Invitae is a genomics, clinical genomics vendor, the turnaround time will be a couple of weeks.
That's pretty fast. So this is great, because I remember actually getting actually it was with Invitae, but it was through our institution and having a genetic counselor at our institution. But this is awesome that people will be able to access a genetic counselor through Invitae and have that whole experience. And the timeline is pretty quick and at no cost. That's awesome.
Mm-hmm. Yeah, we're really excited and interested too, because these results will then also be shared back with our team and the projects. And so we will be able to have, sort of this yet another data point about participants, thanks to their participation in the project. And also, again, want to call out the regulatory restrictions for Invitae only apply to folks who are in Canada in this case. All right, so then the other elements that are new and upcoming in the next couple of months. So we're adding some more surveys to the project. Like we said, a way of continuing to engage with the project, continuing to give us rich information that we know that we can only get from patients and family members. The first one up will be a survey around the family history of cancer and understanding like someone's family tree and the incidence of cancer in different family members, what types of cancer, how old were those family members, just to give us some more information to be able to package and have available for researchers. And we're also taking a keen eye to how we're engaging patients. We're doing it for the project, but how we do it in and of itself could also be a science. We're not the only ones doing this, patient engagement in cancer genomic research. at Count Me In. There's lots of sites and lots of institutions across the US and also across the world that are trying to do this. And so we're trying to take a closer look at how it is that we can be successful. And even the areas where we're not being as successful in reaching patients and engaging them. So we'll have team members who may reach out to folks who are participating in the OS project to ask about your experience, about why you were participating in this, what drove you to participate in something like the Osteosarcoma Project. What do you like about it? What do you not like about it? And that will help us understand better how to engage in the Osteo Project, and also have probably broader learnings for the rest of Count Me In, and maybe even other cancer studies that are engaging patients as well. I'm really excited about this. And along the way, as we've been building these new things, we have been building this, as always, we like to do, with input from patients and family members. So as we built these new elements of talking about receiving results and shared learnings about tumor samples and germline samples, we've heard some really important key points of feedback that we've incorporated, and just wanted to highlight this both as a way of saying, as Corrie was saying, we've been really thoughtful and we've really taken our time with this, but also to know that like the feedback that we hear from the community is really important and we take it very seriously. So some of the things we've done is make both learning about tumor DNA and learning about germline DNA totally optional. Participants get to choose whether or not to participate in this. It could be sensitive. You may not want to know. You may just want to contribute to research, or you may be really interested in getting those details. We've also, for learning about tumor DNA, have been using the term shared learnings and sharing learning and information back with participants over the term return of results, because return of results has kind of a certain weight to it, a certain expectation around the turnaround time, and maybe even the potential impact of, oh, these results will mean something, for my treatment and my care. And because of the state of research in osteosarcoma today, we can't make any guarantees about that. And also it may take us many months as well to get medical records and tumor samples. So we, have shifted things and decided to say, we're going to share what we're learning with you. And we're also trying to be really clear about, what we're finding and have more transparency from the Osteosarcoma Project. So what we know and what we don't know about those variants that we may find in the tumor DNA in those shared learnings. We're also, talking about how these are, yes, will be clinical grade results, which is the reason why we can share them back with participants. But as Corrie mentioned before, first and foremost, we are a research study. And so this is on a research timeline. And that's unfortunately why things will take a good amount of time for us to be able to share that information and those learnings back with folks. And as we keep going, and we're doing this for the first time, we're thinking critically about how we can provide support and resources for understanding this information. So when folks have questions, who can they go to? Especially given that we will be returning, or we will be sharing this information directly with participants, and there's not a clinician or a care partner involved. And so how do we make sure that we are giving folks a place to go to, or a source of support to understand questions that may come up when you are learning more about these different sources of DNA. And so, at the end of the day, like we are very open. We always want to hear from the community more. And so even though we've, incorporated feedback as we've been building these new things, we are an organization that is always open to change. And so we always want to hear from the community. So if you are a participant, a family member, just someone in the community, and you've got a thought to share that could make the Osteosarcoma Project better or different, please always reach out to us. Let us know what you're thinking. We would love to hear it. And I'll just close by saying we have an enormous team of people behind all of these things. You're seeing Corrie and I's faces here, and also maybe in the photo, yes, in the photo as well. But there's a lot of thought and people's energy that goes into making the Osteosarcoma Project and making Count Me In possible. So just wanted to throw all these names up here and say thank you to each and every one of them as well.
That's fantastic. Thank you. Obviously a lot of effort into making this a successful project, and everyone feel free to put any questions that you have in the Q&A, and I just have a couple, Taylor. So I know you talked about the germline testing, and then I think the slide before that you had talked about sequencing data. And so just to clarify, how is that different versus something that a patient or an institution might order from another sequencing company, like, you know, a Foundation Medicine or something? You know, is it like a targeted panel? Is it the whole exome or whole, you know, whole genome sequencing? And then also timeline wise, when someone might expect to see that, if you can maybe just clarify the difference between doing it through this project.
Of course. So the germline, just to recap, the germline testing and return of information with Invitae is a targeted panel. So it's 84 specific genes. The turnaround time on that will be a couple of weeks. For the shared learnings and sort of the return of information about tumor DNA, that will be whole exome. So that will be the information that we discover from sequencing the entire exome, which Corrie nicely animated and explained for us earlier. And the turnaround time for that will actually be quite a while. So it can take us many months to acquire medical records, which then tell us procedures so we can identify tumor samples. It can take then time to acquire those tumor samples. And then our team has to process them, label them, and send them over for sequencing. And sequencing in itself actually takes a good number of weeks as well. So folks may be quite a while between signing up for the project and actually hearing from us with any information that we've been able to gain from tumor sequencing. Since we haven't done it yet, we're not really sure exactly how long that will be. And also it can vary so much, probably from person to person, institution to institution. So we'll see, but definitely we are eager to get that process going and see what it looks like and make sure that we're returning information in a timeline that both works for our team and is also like works for project participants.
Right. So for now, at least, participants should think of that as more contributing to research, getting back those shared learnings, but not necessarily like a clinical actionable data that they could then go and kind of inform their treatment plan. And so I was also wondering, I know, because when you guys launched, it was really like 2 weeks before COVID just completely took off. So, and everything went off the rails. And so I'm, you know, with my COVID brain trying to remember, I remember getting, doing some forms. I remember getting the cute little box where I'm pretty sure we returned our saliva samples, but for all these participants who at some point probably did sign up and register, but can't quite remember where they are in the process, is there a way for them to kind of check in and say like, okay, did I release the medical records? Did I send in my saliva sample? Is it, you know, have I released the you know, any slides or tissue samples? Is there a way for people to kind of just see where they are and having submitted all this information?
Yeah, definitely. Well, first and foremost, you can always reach out and ask. Either, you know, our email address is here, info@osproject.org. But with these new features and new opportunities coming in the next couple of months, we are going to reach out to all existing participants in the Osteo Project to let them know about this. And that will be hopefully a great reminder that you know, folks are participating in the Osteosarcoma Project. And if you have questions or can't remember, did I get a saliva kit? Did I not? Did I move in the last 2 years and maybe I missed it, or maybe it didn't make it to me? You know, you can definitely ask us those questions, and we'd be happy to let you know where things stand and if there's any action that you could take to further contribute to the project.
Fantastic. And then I know this question kind of comes up a lot sometimes when you're just talking, because it's, I mean, this is awesome because it definitely seems like a very central place, because the question always is like, I want to contribute, but I just don't know where to send my stuff, right? So this seems like a really great kind of universal central place for everything to go. But there are, you know, other kind of collection projects happening, and just kind of wondering how this intersects with that. So for example, and I will also plug Factor by saying there is going to be a big data panel there that Corrie is going to be on, and we are going to have speakers from kind of several of these other initiatives there. So we'll kind of be able to kind of have everyone share and kind of figure out how they're different or where there are commonalities. But for example, like the Treehouse Cancer Initiative at UC Santa Cruz, where they're, collecting a bunch of RNA-seq data, DNA data. So how does this kind of intersect and work with some of these other databases that are out there?
Yeah, I'll let Corrie take this one.
It's such a good, oh, sorry. Yeah, I was going to say it's such a good and important question. And I don't have an answer for you right now. And I'm actually really glad that you're convening us, because I think step one is for all of us to know what we're doing. You know what I mean? I think, we go into this feeling really good about making transformational change. And so do other people. And it's up to us to figure out where are the points of intersection and how do we work together. As a patient, I'm going to put my patient hat on. As a patient, I will join every single thing available to drive research forward. Even if it seems like the same, there's always going to be differences. You know what I mean? We're going to have an intake survey that might be slightly different than somebody else. And as a result of those key differences, there could be learnings, and so my goal is to make sure that the people on my side with the research side are doing everything we can to make the process as easy as possible. That's one of the reasons we are dedicated to just open sourcing everything, so whatever we make is just goes out, and part of the hope is so that folks that are on, in some of these other initiatives, they can do whatever they are doing, and they can add all of the data we're generating on top of it. And so it's like a way that we're hoping will break down some of the silos by just giving all of our data away for free. That there is a big issue at play when you have rare cancers. When you have the same patient that joins different registries that where sequencing is at play, you don't necessarily want the researchers to combine that data and think that they have a significant thing that is mutated across multiple people when it's just the same person that's in multiple data sets.
Right. Yeah.
That's the first place we need to start as the research community. I think we need to start there. Everything else is pretty complicated, but that's a thing we just have to figure out, so that when folks are combining data sets, they don't combine in a way that leads us down rabbit holes that are just not true.
So I am so glad. I'm so glad you guys are all going to be able to be together in a month and start talking about those things. Thank you so much. This has been so interesting. Thank you for the update. It sounds like there's a lot of exciting stuff going on. And again, I think, Taylor, are you going to be at Factor?
I will not be able to make it this year. I have a very close friend's wedding to attend, unfortunately, but I will be, thinking of you all and definitely wishing I was there, because Factor 2020 was such a special moment to launch the project and get to be, embedded with the community. It was so great.
Yeah, well, I think this June there are so many weddings because of all the postponed weddings, right, for the past 2 years. So I'm glad you're actually finally going to be able to participate in that event. And but Corrie, we will see you at Factor. And I want to thank you both so much for your time today and for spending an hour with us to make it better for pediatric oncology patients everywhere. More information on this and all Osteobites can be found on YouTube, on our website, MIB Agents, and on your favorite podcast place. And if you register for the session, you'll get an email with more information and links mentioned today. And next week, we're talking with Dr. Breland Wilkie, who's the director of the sarcoma program at the University of Colorado School of Medicine. And she will be discussing immunotherapy for osteosarcoma and review what is known about the role of the immune system in fighting osteosarcoma. And she'll highlight current and future directions for research studying immune-based therapies. So thank you again so much, Corrie and Taylor, for joining us. Thank you to our sponsor, BTG Pharmaceuticals. And thank you for joining us on Osteobites today. And we hope to see you next week when we chat with Dr. Wilkie.
Thanks so much.
Corrie. Thanks, Taylor.
Thank you.
Bye.